So to begin, I just wanted to tell you a little bit about Nexcelom as a company. We were founded in 2003 in Boston, in Massachusetts, in the USA. Our headquarters is in this converted red brick mill building that you can see here. This is where all of our development, manufacturing goes on. And we are a company that focuses on automated cell counting and image analysis. We're also a group of scientists helping other scientists to increase their accuracy and save you time. Those are our two main objectives. And one of our USPs is that we provide really excellent customer service and application support for anybody with any of our instruments. So what do we do? Well, at Nexcelom, we specialize in cell counting, cell assays, and image cytometry. So we have a range of really good top quality cell counters, which is our Cytometer range that you can see here. They have different magnifications, different fluorescent capabilities, really to suit your needs. The newest addition to our Nexcelom family was launched last year. That's the Celica, which is a high throughput version of those cell counters. It's really revolutionizing cell counting in high throughput at the moment. It can count 24 samples in a single run, and it does that in less than 3 minutes as well. So that's about 7 seconds per sample. It's very, very quick. And then we have our image cytometer, the Celigo, which is the instrument I'm going to be showing you a little bit of data from today. It's a plate-based, cell-based assay system where you can image in multiple channels and create beautiful images and accurate data as well. So this is Nexcelom's full portfolio of instruments. We work with Immuno Diagnostic in Finland to distribute those instruments. And our company aims overall are to do your existing assays more accurately, to do them more efficiently, and to perhaps do some assays and experiments that you couldn't do before. All of that as well as creating a user-friendly software. So not a piece of equipment that only an expert can operate, but something that can be put in a multi-user laboratory and that everybody can gain access to. So to start, I wanted to mention some more classical methods of producing results from your in vitro cell-based assays. And those are using a flow cytometer. Flow cytometers are great for analyzing multiple fluorophores or multiple markers within a single population, but they do require quite large numbers of cells, quite large volumes of cells. These cells also always require trypsinizing. The whole process does take quite a long time as well, and you can only really do an endpoint assay on a flow cytometer. You can't take multiple time points. And also, if anybody's used a flow cytometer before, you know there's calibration steps, there's maintenance issues, clogging issues, and cleaning issues. They can be quite difficult to use. Plate readers are very fast and they're good for doing kind of bulk measurements, but you can't do that many cell-based assays using a plate reader, and sensitivity can be a problem. And then manual microscopy is probably the most common method I see being used for these cell assays in the lab. Almost everybody has access to a manual microscope, and most people to a fluorescent manual microscope as well, which is great if you only have a couple of samples that you want to look at. You can get a nice high magnification and great images. But if you have any larger experiments that are more than 2 or 3 samples, it becomes extremely time consuming to do your analysis in this way. There's also no quantification within those methods as well. You will always have to take images and then export to an external software such as ImageJ to do your analysis. So it's definitely not appropriate if you are analyzing different samples in a 96-well plate, for instance. So automated imaging systems such as the Celigo can really eliminate a lot of the problems of the other technologies that are out there. It's almost like having a super speed automated microscope that's in a box, but with the addition as well of that analysis software at the end, and that's all built in. So with the Celigo imaging system, you can image your cells within the plates. There's no need to trypsinize anything. You can just image your cells as they are. There's very little maintenance because there's no fluidics within the system, so there's no contamination problems, and it's very easy to use. It's also very fast, and I'll show you some examples of the speed of the instrument throughout the presentation. And finally, as well, compared to flow cytometers and plate readers, you can get, as well as all this data, gathering it really quickly, you can also get these multi-channel publication-worthy images alongside your data as well, which look really nice for presentations and publications. So just to give you a little bit more information about the Celigo specifically, it's compatible with any plate format. So from 1536 well plates to 6 well plates, you can run your experiments in anything. And it's also compatible with slides, flasks, and Petri dishes. It has a really beautiful bright field channel where you can view your cells in bright field without any label, and also 4 fluorescent channels. And one of those features that kind of sets it apart from other similar imaging systems is that the Celigo will capture the whole well with even illumination and even focus across the whole well. So this allows you to count and analyze every cell in every well, which you certainly wouldn't attempt using a manual microscope. So moving on to some specific assays now. So immunology and immuno-oncology specifically are really widely studied areas and rely on accurate in vitro cell-based assays to be conducted sort of consistently and accurately and efficiently. And I just wanted to go over a few examples of these kinds of cell assays done on immune cells. So the first and probably most common one that we see is assays surrounding cancer cell therapy. So where your overall aim is to introduce new healthy cells into a patient body to replace or combat the diseased or missing ones. So a lot of in vitro experimentation is done before that implantation. You might also want to be optimizing your best culture conditions for your immune cell expansion. That can be really important in cancer cell therapy, but also in other areas as well. You might want to look at your different populations of immune cells and do some immunophenotyping, see what proportion of T cells, B cells, monocytes, macrophages you have within your population, and also see if they're activated or being activated by a particular treatment. And yeah, my final 2 are just monitoring immune response as well. And I've put in vivo here because you might be doing work on some in vivo models. You might be applying different treatments in your in vivo models, and you want to isolate the immune cells after those treatments to see if they have produced an immune response. And of course, you can look at that in vitro as well. So you can have your immune cells, you can apply specific drugs to those immune cells and see if they are activated or start proliferating. So with some of those cell assay types in mind, I wanted to show you what you can achieve using an imaging system such as the Celigo. So all of the images and data that I'm going to show now is produced directly from the Celigo, and this first one is a very simple label-free proliferation assay on T cells. So cells are detected and identified without a fluorescent label. This is the whole well image that you can see here. I'm just going to zoom in on an area here, and you can see that all your T cells are there, and we can see that we can accurately identify all of those individual cells as well. This blue segmentation on the right hand side here is just showing you the Celigo analysis software and how that detects each individual cell. This sort of experiment, if you were to do a 96 well plate for this and do different treatments, maybe different media conditions, different supplements in each well, this would take you 6 minutes to scan and analyze an entire 96 well plate. So just compare that to how long it would take if you were doing these kind of experiments manually. It's a lot quicker. And this can be done as part of a time course experiment as well. So you don't just have to take an endpoint reading, you can look at different time points within your experiment. So in this example, each day the plate was placed into the Siligo and scanned, and the cells were quantified in each individual well. So what we have here is we have day 0 results. And we can see that in all of our wells, the cells are very consistent at day 0. They were seeded very well. They all have around 8,000 or 9,000 cells per well. And then in some of those wells, so in rows B and rows C, that's the top 2, we have the addition of growth factor cocktail 1. In the third row down, we have the addition of growth factor cocktail 2, and the bottom row is a negative control with no growth factors added at all. So by day 3, we've been imaging every day, but up to day 3, we can see from this heat map here where the proliferation has been occurring. So in the top 2 rows, that's with growth factor cocktail 1, we can see there's been a lot of proliferation. There's now around 60 to 70,000 cells in those wells. So they've really been proliferating with that growth factor cocktail 1. In the third row down, growth factor cocktail 2, we've seen some proliferation, but definitely not as much as the top, as growth factor cocktail 1. And with the negative controls, very little proliferations happened here. And we can also, what the Siligo will do is it will plot those time points for you for each individual well, so you can immediately see the best conditions for your T cells to be in. And again, every time you scan this plate, it's going to take you, if you have a whole 96 well plate, it will take around 6 minutes to do. So it's a very quick and accurate way of looking at the best conditions for your T cells. So I spoke earlier about the ability to image the whole well, and I just wanted to touch on that point a little more. This is the reason, why is it important to image the whole well for these types of experiments? Well, this is a really good example of that. A lot of slower imaging systems or high content imaging systems will just take a central portion of that well. They'll look at the amount of cells in that central portion, and they will assume that the rest of the well is similar to that central portion. So these 2 wells here, well A, if you only took the central portion, you would say that that well is 95% confluent. We've got a kind of yellow fill over where the cells are here, just so I can show you where the cells are, and where the cells aren't is that grey area. Well B, on the other hand, you can see a lot more grey area in there where there are no cells present. That well is only 40% confluent if you just take that central area as to be representative of the entire well. But as we know, cells don't really grow uniformly in wells. They either gather around the edge or they can gather in the center and grow quite non-uniformly. And if we look at the whole picture of both of those wells, you can see that this is definitely the case. They're not uniform across the whole well. And when you do take the whole well image into consideration, you can see that actually they're both around 70% confluent. So there's no difference between A and B when you look at the whole well. But if you were to only take that central portion, you would definitely say that the cells are more proliferative in well A if you were to just look at that central bit. So this is why for our experiments and for using the Siligo, we definitely suggest you look at the whole well, and that's really important. So in terms of how many cells can you accurately count per well, we have some suspension cells here, and you can get great segmentation at a range of cell concentrations. So we have 8,000 cells here. We can see that the cells are quite spread out. They're very easy to detect accurately. We can go up to 30,000 and we can see they're getting a little more dense, but we can actually go all the way up to around 120,000 cells per well and analyze all of those individually in roughly the same time. So, you can really get a lot of data from a single well. Okay, so another factor you might want to monitor is cell health throughout your process. And there are ways you can do this kinetically, or you can do this as an endpoint experiment. So not only do you want to look at proliferation of your immune cells, but you might want to see the health of those as well. So there will be times when you might be optimizing the right media, you might be optimizing freezing and thawing conditions and what gives you the best T cell health. So you want your cells to be super healthy and ready to be used as part of your experiment. So this example here, the green stain is for caspase 3,7, which is an apoptotic marker, and the red stain is propyridium iodide, a marker of necrotic cells. So we're looking for the effects of these different media conditions. So between medias 3 and 4, you can definitely see that there's an effect on necrosis with media condition 4 here. So you will be less likely to choose that media condition for the sake of the health of your cells. Identifying the various populations of lymphocytes you have present in a sample can also be an important assay. So what we have here is an example of immunophenotyping. So we have a mixture of red blood cells and white blood cells. In this sample here, you can't tell the difference in the bright field image. It just looks like all the cells are the same. But as soon as we start applying different antibodies to different markers, we can then see where the white blood cells are. So CD45 is a panleukocyte marker. So we can see where the white blood cells are within that population. And then more specifically, we can use CD3 conjugated to Alexaflor 488 for the green signal. So that's for your T cells specifically. Then we've got CD19 for your B cells and CD14 for your monocytes. So this is all within the same sample. We can merge those images together and we can see which of those cells are T cells, which are B cells, which are monocytes, and all of that can be quantified. The population analysis, you can go one step further and export all of that data into sort of flow cytometry software. You can do that really easily. And then you can do even further population analysis. So this is looking at the proportion of T cells within your sample. And then we can also compare CD14 to CD19 and look at the proportion of monocytes and B cells as well. And all of that can be automatically quantified for you. And then you might also, this is my last experimental example for the immunology assays, you might also want to look at activation of your immune cells. And it doesn't always have to be through looking at a surface marker or cytokines. It can also be through simpler methods like looking at proliferation, cytotoxicity, or like this example, which is by monitoring the size of those T cells. So what we can see here is at day 0, the T cells are quite small and rounded, but then by day 6, we can see that the morphology is changing and they're definitely a lot larger. And then we slightly see a reduction in size of those at day 10 as well. So we can kind of see where the activation is occurring. Okay, so at this point, for the second half of the presentation, I just wanted to focus on some virology cell experiments as well. And, you know, considering the pandemic we're currently experiencing, it's relevant to relate COVID-19 to the different types of virology research being done at the moment. So we're just talking about the virology research in the labs. We're not talking about clinical trials, just things that are being done in the research labs at the moment. The areas of research that everybody is still talking about, even now, sort of a year and a half in, are screening of antiviral drugs and vaccine development. We're seeing new vaccines being developed all the time. So all of this research starts in the lab. But it's important to also consider those other areas that are being spoken about as well. So studying the mechanisms of pathogenesis of the virus in the lab. If you look at those mechanisms, that can help then determine what would be good candidates for vaccines and antivirals. But then we've also got the diagnostic testing that's been widely reported. Evaluation of the preclinical models, so looking at the infection in some animal models, that's also done in the lab. And coupled with all of that research, we're also looking at our immune response to vaccines, to antivirals, and to the virus itself. So there's lots of different areas of research going on in the labs at the moment. And actually the specific types of assays being performed are quite varied. So we've got plaque assays being done. I'm sure no matter what kind of lab you're in, anybody doing virology experiments within your institute, you'll see these kind of plaque assay plates piled up on their bench. We've got focus formation assays where we're using a fluorescent dye to indicate those little focus formation areas showing you where infection is happening. Cytopathic effects, so just looking at cell death within your sample. Then infectivity, so just looking at viral infection within your sample with different conditions, maybe looking with different antibodies to try and neutralize that infection. Those kind of assays are very popular. And finally, just cytotoxicity. If you're looking at different drugs for treatment, you want to see the cytotoxicity of those drugs as well. And looking at proliferation and monitoring morphology changes within your cells after infection as well. Lots of very varied experiments going on in the lab. So I just wanted to talk just about a couple of those, just give you a couple of examples of the types of work that people are doing and how they are streamlining their efforts to get as much research done as possible, especially when it comes to looking for answers to COVID-19. This is a group who are doing infectivity assays. This is probably the most common type of experiment I see when I visit virology labs. And the experiment we have here is with human cytomegalovirus, so HCMV, and it's on fibroblast cells in a 48-well plate. The virus is detected using a GFP antibody, and our host cells are detected by using a DAPI counterstain. The output that they want is the percentage of cells that are infected across the plate. And what they've done here is they've used different viral mutants per row. So all of those rows, so A, B, C, D, E, and F, they're all different mutations of the HCMV virus, and they're looking for potential vaccine candidates. If you were to try and do this, this experiment on a flow cytometer, just one single plate, what you would be expected to achieve, 48 samples, 10,000 events per sample. We're talking about the cleaning, the setup, and the analysis at the end. I think you'd be quite happy to complete that experiment in around 2 hours. Another way of doing it would be through manual microscopy, taking images manually, analyzing those using external software for your 48 samples. Again, you'd probably set aside a whole day to complete this 48 sample test if you were taking all those images manually. So with those timings in mind, I just wanted to show you what you could do using an imaging system. So using the Celigo, we can detect the total cells with the DAPI, so that you can see here, and then determine which of those are infected by looking at the green GFP signal. So what we've got here are different viral dilutions. So from left, we've got high amount of virus, and then we've got a low amount of virus on the right-hand side. We can see the difference of infection rate just by looking at the images. But then we can also get the results from the Celigo as well to show you the infectivity rate at those different virus concentrations. We can draw infectivity curves for each of those virus mutants. This was just done by exporting that data into Excel and just drawing those curves. And the total time taken to complete this experiment was around 8 minutes using the Celigo. So you could run around 15 plates in the same time, taken for one on the flow cytometer, and you could do a huge screening experiment of sort of 40 plus plates in the same time as it would take you to do one by using a manual microscope. So the amount of time you could save if you have access to one of these systems is really amazing. And a great example of this type of screening, with lots of different plates that they're running through the Celigo, is happening in relation to COVID-19 as well. So this is a paper from a group at Sanford Burnham and a collaboration with Mount Sinai as well in the US, and they're performing drug repurposing screens for COVID-19. So they started with 12,000 compounds and narrowed that down to 17 with a combination of using the Siligo and some other methods as well. And then they can take those 17 candidates forward and do some more in-depth testing on those. So take a look at that paper if you want to see how that's working. So some viruses form foci or clusters of infected cells, and it may be the number and the size of these foci that are interesting rather than the total number of infected cells like the experiment that I've just showed you. So a closer look at one of these experiments shows how we can detect those individual clusters or those individual foci at different virus concentrations. So we've got virus concentrations that are high on the left, going to low on the right. And you can see the difference in the number of those fluorescent foci. So data from this can be exported into an Excel file, and you can draw your viral titer curves. This was done on influenza. So yeah, all of this data is generated very quickly. The main point for this particular experiment and for the group running this experiment is to compare their previous workflow to their current workflow. So this is a real life lab using the Siligo now. They previously had to do everything by manual microscopy. They wanted to take 100 images per well and then manually count all those individual foci per image and then write those down in an Excel file. It would take them a day to complete a single plate. Now they have the Siligo, it's all automated, everything's done automatically, the data matches exactly what they would get if they were to do it manually, and it takes them 20 minutes. So they can now have the capacity to run 20 plates per day using this method. So the final type of assay I want to talk about today is the classical plaque assay, which I'm sure everybody has seen in one form or another before. It's still most often used for vaccine development and identifying antiviral compounds, and it's made by adding different quantities of virus to a monolayer of host cells and waiting most often days for those plaques to develop. When the plaques have developed, your cells can be fixed and stained with crystal violet, and where a plaque has formed, we will see a hole in that monolayer. So this is an example of one of those experiments. We have Vero cell monolayers, which are infected with a virus and then incubated for 36 hours. The monolayers were then stained with crystal violet, and we're quantifying the total number of those lytic plaques. It was previously done manually by eye. And if anybody's done that before, it normally looks something like this, either on a light box with a marker that you put a small dot where all the plaques are, and that's how you count them, or hold them up to the light. And the time taken to do that manually is around 1 minute per well, which is pretty quick. It's not a slow process. It's quite efficient. However, there are some drawbacks in this process. There's a lot of user-to-user variability. You know, is it a plaque? Isn't it a plaque? Is that one big enough to be a plaque? Is it not? Depending on what user counts those plaques, you can get very different results. You don't get any data on the size of your plaques. There's no images being created. And you also need to wait until those plaques are big enough for you to be able to see by eye, which can sometimes take, this one was quite quick, but can sometimes take between 7 and 10 days to even appear. So these images were taken on the Siligo. It also takes around 1 minute per well to do this analysis on the Siligo. So there's no real time saving in that aspect. But there are other advantages and other ways of streamlining this kind of assay. So we can see from these images, if we zoom in a little bit here on the more concentrated image, we can see that the segmentation of the plaques on the Siligo software, that's really good. The Siligo does a great job of quantifying those plaques. The green ring around those plaques that you can see on the right-hand side shows you that even when the plaques are merging together, the software does an excellent job of identifying whether it's 1 plaque, 2 plaques, 3 plaques, etc. You could argue that you could just, by eye, you could just use a lower dilution of the virus to get an accurate result, even when there's a lot of merged plaques there. So we've got well B2 for that. But even at the lower concentrations of virus, you still get plaques that are merging together. With the Celigo, you can zoom in and you can really tell if this plaque here is 1 or 2, and it does genuinely look like it's 2 plaques. That's identified correctly on the Celigo. But if you were looking at this manually, I think you would really struggle to determine whether these were 1 or 2. So in addition to that on the Celigo, we can also consider an earlier time point, again, where you can zoom in and you can see those really small plaques. These wouldn't be visible by eye to you, but they are visible on the Celigo. And I'll just show you a zoomed in image there. The green fill area here isn't fluorescence. This is the Celigo software just showing you that there's a plaque in those locations. So on the Celigo, we can see these plaques. By eye, you can't. And this is a whole day earlier than the previous images were taken. So to summarize this experiment, the Celigo enables you to detect the plaques a whole day earlier than you would have done if you were doing it by eye. So you're not saving time when you're actually counting those plaques, but you're saving a whole day in terms of your experimental time. So that's another way to streamline. On top of that, you get extra data as well on the size of those plaques. So for some people, it's not just about the number of plaques, but also the size of those, which is indicative of how quickly the plaque is spreading. And if you wanted, again, if you wanted to see some published data on this, then there's a publication from Biogen. It's a really good place to start. So Biogen are a gene therapy company in Boston who are now automating all of their plaque assays using the Celigo, and that increases their speed, it increases their throughput, sensitivity and robustness, and gives them that extra data as well. So when we talk about streamlining, you think about getting your results earlier and finishing your project faster, which using an imaging system would certainly do for you, but that's not all you get. You will also be able to do more replicas, get more data, and get tighter data by you doing more replicas. For the virology assays, you will be able to miniaturize your assays into smaller well plates and therefore use less reagents, less cells, and less virus. So it can be a lot cheaper in terms of reagents. You will eliminate any user variability, and you can digitally store all those plate images as well. So you have all these beautiful images in storage if you ever need to use them for publications or presentations. So yeah, that was my last slide. So thank you really very much for your time and attention today.